let's talk about the Digestion Lab a little bit. Most people came up with, wrote their pre-labs, and most of them look pretty good. I'd say one place where people had some issues was in the just objectives. This is one of these labs, since it's three separate actual labs, it actually makes sense to have, it's just easier, I think, to do three separate little objectives, right? For, part one, what is our independent variable? What is our dependent? Oh, it's probably easier to, what is our dependent variable? Or dependent variables, actually. What are our independent variables?
Is it the salivary amylase?
So what would that be?
The independent, and is the breakdown of carbohydrates the dependent?
So one is going to, so independent is going to be presence of that salivary amylase or enzyme. That's one of them. Dependent variable will be, breakdown of the carbohydrate, and as this will ultimately be measured by these two different tests, Benedict's and Lugol's. Are there anything else we're varying besides the presence of salivary amylase?
The pH of solution.
Yeah, the pH as well. So we're looking at how the presence or not of salivary amylase, as well as how pH affect the breakdown of carbohydrate, which we're going to see by looking at our presence or absence of starch or presence or absence of sugars appearing. So for part two, what are our independent and dependent variables?
Presence or absence of pepsin and pH.
Exactly. Again, it's the enzyme and the pH, and the dependent variable is going to be the breakdown of the protein, as evidenced by the shrinking size of the little peps of the egg square. For part three, what is going to be our independent and dependent variable? This gets a little different.
Dependent variables would be the presence of pancreatin.
So independent here.
Oh, I'm sorry, independent, thank you. In the presence of bile salts.
Yeah, so here we're varying, we're not doing anything with pH. We're varying the enzyme, and then we're also varying the presence or absence of bile salts, which are, we talked about, emulsifying. And then dependent is going to be, breakdown of the lipid, as evidenced by it turning pinker based on the presence or the appearance of fatty acids that show up when you break apart triglycerides. Right, so that's, it's pretty simple when you think about it that way. Bonnie?
Would another independent variable have to do with it being an aqueous solution, or like with the how it reacts with the water? Or?
No, not really.
Okay.
Yeah, I mean, pretty much everything in your body is in an aqueous solution. It's kind of a given that everything in the body is happening in an aqueous solution. So that is kind of the big picture. We're looking at these three things. Normally what you would do is set up a whole bunch of different tubes and then you would incubate them, and then you would test. Obviously, we're not doing that in person, but to get a sense of how this works, so this kind of feels like those old things where this feels kind of like when you have like the lazy junior high school teacher that doesn't feel like teaching and just shows the kids a movie. So I think what we're going to do is I'm going to show you me and Tina and Becky kind of going through these processes so you can see how we got the data. Then we have pictures of the actual test tubes that you will use for your actual analysis as your results and for your discussion. But I do believe that those test tubes don't really make sense unless you actually see the process that we used to get them. And also part of the first, in the second part, no, I'm sorry, the third one, part 3 also has another piece where you are just using oil and water with bile salts to kind of get a feel for emulsification. So that you can't see unless you watch the video. And there's also a thing on part 1 with the carbohydrates where we talk about the saliva having actually 2 parts. So maybe I should actually, oh, what the heck? All right, it's back. My cat somehow turned off all my pad. All right, it's all good again. So saliva made by your, the parotid salivary glands and submandibular and sublingual in going into your mouth. Saliva has 2 main components. Part of it is actually just mucus, right? You think about saliva being kind of icky and gucky, and this is going to help bind your food into a bolus. Right? If you chewed up a bunch of crackers and tried to swallow them, it would be bad. You'd just be trying to eat dust. It would not go down your throat. The mucus in your saliva helps bind all the food into a slippery ball. Bolus just means ball that will then slide down your throat easy. But the mucus is not going to be about digestion, breaking things down. So saliva also has, it's called serous fluid with amylase. Amylase is actually a more generic thing. That means things that break down starches. Salivary amylase is actually called ptyalin, but it doesn't matter. You can just call it salivary amylase. This is the part that's actually going to break down the starch into simpler sugars. So part of the lab will actually show, hopefully give you some evidence that, oh, both of these are happening. We're going to have evidence that something in saliva helps break down starch. And we'll also have some evidence that something in saliva is goopy and slippery. So that'll be part of the little videos as well. All right, let's do this. All right, so this now exists as doing the lab, digestion. Then once we go through the kind of doing the lab, I'll go into the results section so you can see close-ups of the test tubes after we're done with everything. All right, here we go. So this first part is going to be the setup of the carbohydrate.
This is Lumen, the first device for hacking your metabolism. With just one breath, Lumen tells you what you're currently.
All right, so this first part is going to be looking at kind of the setup. Remember, there's going to be 5 test tubes. And this is kind of just showing you how that all fits together.
Okay, we're going to be going over part 1, salivary digestion of carbohydrates. And the enzyme we're using for this part of the digestive lab is salivary amylase. And the source of that is our saliva. So it says to collect 7 mils of your own saliva in a graduated cylinder. So I have close to 8.5, it's 9 mils. And so I've been elevating, collecting it in my little spittoon graduated cylinder. We're going to be using this for not only the spot plate experiment, but also for the 5 tubes of the carbohydrate digestion.
So here again is my lab partner saliva so this is going to be kind of part 1 of that carbohydrate thing. I don't know if people read, if you have it in front of you, where we're taking some of the saliva and adding in some vinegar, acetic acid, which actually precipitates out the polysaccharides, the mucins that make saliva kind of gunky and sticky and slippery. So this first part, this is part 1 of steps 1 and 2 of part 1, which is where we take some saliva and put it on a little spot plate and add this acid. And so we can see what happens. Again, this is going to, you'll have in your lab write-up, you need to describe what you see here, because that's part of this lab, is understanding that saliva has 2 things going on. And part of it is, you're going to see here with these mucins. We were using as our source of the amylase enzyme for the carbohydrate part of today's lab. But saliva has a couple of roles. One is helping break down carbohydrates when you're initially chewing up your food, but it also has mucus that helps kind of bind your food into a bolus to make a nice little slippery ball you can swallow down your gullet. And so we can actually see that other aspect of the saliva. If we take some saliva here, just taking a mil or so of saliva, and this is acetic acid, is basically kind of diluted vinegar. Right, so that's just Becky's spit right there. And we're going to add in a little vinegar. We'll actually precipitate out the mucins, the mucus part of the saliva. So now use a toothpick. We can see, like, ooh, look at that, all those gooey, goopy strings there. Those are the mucins that are the other component of your saliva that are helping kind of bind and lubricate your food. So imagine trying to swallow a bunch of cracker dust. Yeah, it wouldn't really work. So this is what makes a nice slippery ball go down your esophagus. In fact, yeah, if you look in detail, totally cool gooey goop in there.
The anesthesia.
So that you get to see, again, it's a polysaccharide. The actual structure of the mucins that's actually, I met a woman recently at a conference who studies this as her main focus, because I mean, it's a key part of physiology, and it's a molecule that we still don't totally understand or exactly how it's put together. Like making fake mucins is not something that we can do so easily, even though you might think it would be a really more straightforward thing. Some things that seem they should be easier or not. So that is the first part. So the rest of the carbohydrate part 1 is going to be using saliva in its other role, as an amylase that's going to break down starches. So the next part here, what time? Yeah, this is, so you're going to just sit right back in here a tail of setting up a bunch of test tubes with different amounts of saliva and water and different pHs and saliva, and we'll see what happens. So this is like 13 minutes. So here we go. All right, so now we're going to describe part 1 of the Digestion Lab. Here, the salivary digestion of carbohydrates. We're going to be using this saliva that was generously provided by my lab mate. And this will be the enzyme. Then we've got our substrate for the enzyme, our starch here. And then we can look at how this process happens under different conditions. So to start, we're going to have to just, and I should just, there was some misunderstanding in some people's pre-labs. So starch solution, it's starch floating in water. Does starch spontaneously break apart if it's just sitting in water? No, it's like just a molecule that stays intact. You need some kind of enzymatic degradation to break starch apart. Starch doesn't just spontaneously start disassembling itself. So this starch, if I just leave starch as starch solution, it's just going to stay starch solution. Just put some starch. We have 3 mils of starch that we put into each of these 3 tubes. Just gonna here. Here we go. There's 3 mils in there. 3 mils in there. 3 mils in there. And then also 4 and 5 are going to be controls. Part of an experiment, whenever you're using some kind of an assay, you want to validate your assay. Make sure you give your assay something where you know what the answer is supposed to be, and make sure you get the answer that you're expecting. So then you trust what the answers are when you're looking at your unknowns. So for test tubes 4 and 5, we're going to have test tube 4, which is just going to have pure starch in it. And when we do our test, it better say that, oh, it just has starch in it. Test tube 5 is where we're going to have pure maltose. I might as well just put that in right now. I will put that in a little later. And so when we can check our assay for maltose, that better say pure maltose with no starch. Right, so this idea of validating your assays is really important, because otherwise, if there's, how do you know to trust the unknowns where you don't know what the answer is supposed to be if you don't actually totally have confidence that your test is telling you what you think it tells you? Right. Again, I think I've mentioned one of the classic examples was some of these early scaring studies about LSD is breaking your chromosomes. And, somebody did some experiment, and according to their test, LSD was breaking chromosomes, and they published it, and they eventually had to retract it because when people looked at their methodology, it turns out that if you use the exact same method and you added milk, the milk broke all the chromosomes in the test. It turns out when you use tap water, the tap water was breaking chromosomes in their test. So it was obviously not a good test to see if something was like a mutagen. So you really want to make sure, if you have some test, put something in it where you know what the answer is supposed to be, and make sure that it's giving you the answer you expect, because that way you have more confidence that it's actually telling you what you think it's telling you. All right, so we have starch in our 3 unknown tubes here. Test tube 1 is going to just fill out our volume by adding 3 mils of water. One. see this thing, 3 mils of water. We got 3 mils of water in test tube 1. Test tube 2, we have to add 3 mils of this quality high grade saliva. And that's gonna bring this up to 7, so we spit it out. We're gonna spit it up into there. For test tube 3, before we add the saliva in, we are going to increase the pH by using this concentrated hydrochloric acid. This stuff is something you got to be careful with. You should not be using this without wearing eye protection. You know, regular glasses can work. If you don't wear glasses, you got to be wearing goggles of some sort. This can damage your eyes, so, and your skin or your clothes or anything. So be careful with it. We're gonna add 5 drops of the concentrated hydrochloric acid here. 1, 2, 3, 4, 5. And sure, so now we have this very low pH solution, and now we're gonna add 3 mils of our saliva into this. So 3 mils of saliva, but the conditions here are going to be very acidic for this salivary amylase to try to do its job. Why do you think I made sure to put in the acid first and then put in the saliva?
If you did it in the other order, some digestion would have occurred before you put in the acid.
Exactly, and we'd have some saliva at regular pH having a chance to, so then we wouldn't have just a clean result of what is happening to the saliva activity in acidic conditions. So, but because we did acid first, we know that the saliva never had a chance to see anything other than hyperacidic conditions. It's important to remember, keep it in your mind now, that each of these tubes, we're going to be testing both for starch using iodine, as well as testing for the presence of the products of digestion, maltose, which we're going to use Benedict's reagent for. So we're going to be putting these in the incubator. And after we're done, though, we need to make sure we take each of these tubes and we split it into 2 tubes. That way we can do the Lugol's test to look for starch in one set, and we can use the Benedict's test on the other side to test for the presence of maltose. If you forget to do this and all of a sudden put your Lugol's iodine in here without having split it ahead of time, then you're not going to be able to do the test for maltose. So at this point, we're going to take our tubes here and we're going to have to incubate them at body temperature. And we'll come back in a while and see what happens. So here we see test tubes 3, 2, and 1 are joining the test tubes from parts 2 and parts 3. And we're gonna close this puppy up and come back in a half an hour or so and see what happened. All right, so these have been incubating at 37 degrees C, and now we can do our assay and see what the results are. All right, so each of these needs to be split into 2. Right, so this is now, it's had a chance. Whatever's going to happen's happened, and now we're going to, again, do both assays. Test for starch with Lugol's, Benedict's for maltose, which is basically the breakdown parts, the sugars, simpler sugars, disaccharides. 2 roughly equal test tubes, so we can check each sample both for maltose as well as for starch. And again, it doesn't matter exactly, it should just be roughly equal. And these are my 3 experimental tubes here. And then remember, I also have the control tubes I have here. This is just pure, 4 was pure starch. So when we do our test, this should show up positive for starch and should not show any maltose. This one here is pure maltose. So we do the test for maltose. This should be positive for maltose, but have no starch present. That's how we're gonna know our test is trustworthy. So we can start with the test for starch. We use this iodine, and how many drops do they say? 3 drops of Lugol's iodine. So I'm going to put 3 drops in each of these tubes. 1, 2, 3. 1, 2, 3. 1, 2, 3. Wow, that's not good. Oh, that's good. 1, 2, 3. 1, 2, 3. All right, so if we look for starch, 1 is dark purple black, presence of starch. 2 is amber, that's just the color of the iodine. So this would be starch, looks like. 3, we also have a very strong reaction here. And this is encouraging. Our pure starch is showing positive and pure maltose is showing negative. To do the maltose test, we have to. So does that look good so far? What is that kind of what we would have expected? Yes, no. I don't hear anybody.
Yes.
Right, so it's number 1 was just the starch and water. You know, just keeping it a little warm doesn't make the starch is still there. It hasn't fallen apart. Did the saliva seem to make a difference?
Yeah.
Yes.
Yeah, big time. If you take a look, this again, this kind of more amber brown is just the color of the iodine. If you compare 5 where we had absolutely no starch, it's pretty much the same color. This is, what is this telling us about what happened in test tube 2?
Some of the starch broke down.
Pretty much all of it.
Maltose, pretty much all of it.
Right?
I was a little confused. Would it be red then if all the starch broke down, or would it still stay that like reddish yellow?
Right. No, those other colors are for Benedict's. For the
Okay.
iodine, it's just this kind of yellow brown, just as
Okay.
nothing's going on, and kind of more of this blackish purple if there is starch. So in 3, did the acid seem to make a difference?
It made a difference. It didn't result in any of the breakdown because it prevented saliva from making a difference.
Right, so it made a huge difference compared to, so saliva in this really low pH did not do anything, right? So that's important to notice there. And then again, our controls are giving us what we were hoping. You know, starch shows starch, no starch shows no starch. Next, we are going to look at the Benedict's test, which is looking for the breakdown products of the digestion. To do the maltose test, we have to add 4 mils of Benedict's solution into each of our other tubes. So now I'm gonna, again, for each tube, we'll get 4 in there. Pour in there. And finally, all right, so now each of these is gonna have to go and get boiled for 2 minutes. So let's put these in. We have this in boiling water here. We could also make sure, like when you're using a Bunsen burner, make sure you have a nice blue flame. That's hot. If it's not adjusted, this is not hot. The water's not going to boil. You want to make sure you have the blue cone of heat. So again, just to show you again with the starch, pure starch is going to have this very strong reaction where you get this purple black. And in the case of the maltose, where there was no starch present, we can see the iodine just stays this kind of amber color. So this was no starch at all. This is showing presence of starch. And we'll come back in a couple of minutes when the Benedict's solution has had a chance to do its magic. All right, so these have had their 2 minutes in the boiling water. I'm gonna carefully put them back in the rack here so we can see what happened. These are controls. Three. One. There, we can turn off the fire. And now let's go take a look. So first we can go to our controls again, and we can see this Benedict's is blue initially. It stays blue if there is no evidence of maltose or any of these reducing sugars. And so this is what we were hoping. This was pure starch in here without any maltose, and this shows from Benedict's that there's no maltose present because it's staying just blue. This would be a very strong positive reaction with Benedict's where it turns this reddish orange. Again, there's a whole range of colors it can turn. It can turn from kind of greenish all the way to this kind of tomato red orange. And that would all imply the presence of maltose in our case here. So this is great from our control perspective. Starch was negative for maltose, maltose positive for maltose. If we look at our unknowns, this was starch just with water. So starch basically on its own after sitting at 37 degrees. Blue means, what does it mean? And why? 2, this is showing positive for Benedict's. We definitely got maltose appearing in this tube. We can talk about that in your discussion, why that makes sense. And 3, this one was the same as 2. These were both exactly the same, except in 3, we had that concentrated hydrochloric acid in ahead of schedule before we put in the amylase. So the enzyme ended up having to try to do its job under this really low pH condition, and we can see a very different result. Definitely something you want to think about and discuss. Okay. So that is part 1. So there you've got to see what you would have done, but it also gives you a sense. Like when we get into the results, I have pictures of what the tubes look like for, Lugol's versus Benedict. So you can use these, you can print these out and use these as part of your write-up. Also in your lab practical, your lab exam, that's going to happen in a week and a half. It's like on the last day of official day of class. Part of a lab practical is knowing, how we measured stuff. So you should be able to understand what these pictures mean, what the significance of these colors are, and what they tell us. You shouldn't just know this lab intellectually. You should also know kind of the practical ways, like how did we know that starch was there or not, or sugar was there or not? So make sure that when you look at these pictures, you understand what these colors mean and why, how this helped give us information that we cared about. So the next part is going to be, so I should say, any questions about that so far? Well, we can do a wrap up in the end once we go through all the procedures.
I had a question about tube number 3 for the Benedicts. It
Huh?
looked like it was like cloudy. I don't know if that was just because it was like mixed up, or I don't know if that's something we need to know.
Let's take a look.
Because I predicted that that tube will stay blue since the enzyme to break the starch down would be denatured by the hydrochloride, but okay, maybe not. Or yeah, it seems like it's a little.
Right, but it's not changing color from blue. I mean, I think just
Yeah, so the colors are more important.
right. Yeah, the saliva has kind of slight, it's not as totally glass-like transparent as water would be, but the color is not changing anything from blue. Maybe it's a little less transparent because of the murkiness of the saliva.
Okay, thank you.
All right. All right, so here we go. 4 minutes and 44 seconds of egg whites.
All right, we're gonna go over part 2 of the Digestion Lab. This is the gastric digestion of protein. So as you look at the instructions for the lab, the first step is to using a razor blade to cut 5 uniform thin squared slices of cooked egg white. It's important to make sure that these slices are the same size so you can compare the relative digestion. And when we assess the level of protein digestion, we're going to look at how the size has changed along with possibly a couple other factors. So these are uniform. So how these little egg white squares were made was after an egg was hard boiled, the egg white was sliced into very thin sections, put on this graph paper, and then the squares are the same size as the little squares in your graph paper. So if you put a thin slice of egg white, line it up, and then just with the straight razor to go ahead and slice aligned with the squares, and you have the 5 uniform squares. So each of
Right, because it is important to be kind of pedantic and anal about just making them look really good, because you're not going to really be able to compare the size differences unless you really know that they all looked exactly the same to start. If one's a little smaller later, it's like, how do I know that I just wasn't a little sloppy and it was a little smaller to start with? You make sure that they're all perfect or perfect enough.
those is going to go into each of one of the 5 test tubes that we are using for this lab. So it'll get in there when we add the various other chemicals. So as you can see from your lab book, we have 5 test tubes that we have labeled 1, 2, 3, 4, and 5. And the egg white is going to go in each of the test tubes, along with the different combinations of other chemicals. So test tube 1 is going to have 5 mils of pepsin and 5 mils of water. So each test tube will have 10 mils of different combined solutions. So here is pepsin. And you can see that pipetting 5 mils into test tube 1. And then 5 mils of Bi water into test tube 1. Right? So then test tube 2, it'll have 5 mils of 0.5% hydrochloric acid. Tube 2. Along with 5 mils of water. All right, that's test tube 2, test tube 3, and the last tube, test tube 5, will consist of 10 mils of Bi water. Should be thinking what test tube this is trying to represent. So we're going to put these tubes in the water, the warm water bath at 37 degrees Celsius. And we're going to put them in for at least a half an hour. And if you can allow more time, an hour is better to assess the protein digestion. So we'll see that later. So go in water bath, set the time, and retrieve them in about an hour.
All right, so that was the setup. What I can say is when we looked at those, you'll notice that there is not a picture of them. We didn't like have a little video of what those look like afterwards. They ended up all looking pretty much the same when we were done, which is not what hopefully you have predicted or what I was thinking. But in your write-up, it's something that is, it's worth mentioning what kind of things, because things like that happen, things go wrong. And that means that first you have to decide, was it my hypothesis or is it something about the methodology? But just to kind of put that out there, like things don't always go right. All right, so the last part, we have lipids. So if you remember, I don't know if you remember, if you have your your paper in front of you, there's two parts. There's the part, step one, where you actually are just looking at the emulsifying properties of bile salts. Like, normally what happens if you mix oil and vinegar to make your salad dressing?
They separate.
They separate pretty fast. But when you emulsify, if you can actually even adding in some mustard or something, or if you buy like, Newman's own like balsamic vinaigrette or something, you mix it and they stay mixed a lot longer. And that's because they have these emulsifiers in there that help the oil droplets stay separate and floating in the water and not separate as fast. So this first part is just looking at what happens and you mix up oil and water with or without bile salts, just so you can really see what they're doing just kind of on their own before we start putting them in the context of fat digestion. So this is part 3, step 1.
All right, so here I'm demonstrating part 3 of our digestion lab. And the first part is to investigate the properties of bile salts and how they can emulsify water and oil. And so here I have 2 test tubes labeled A and B. I have added 3 milliliters of water and 3 milliliters of oil. And so the water is at the bottom and the oil is on top. So both test tubes have the same. What I'm going to do now, I'm going to add some bile salts into test tube B. And so you just need a little pinch.
Right, so this is also like in your write-up, you should describe the results here. Like in part 3, step 1, it asks you to do this and describe. Obviously, you'll watch Tina do it and describe what you see in the video. Like what happens when you mix the oil and water with or without bile salts.
And then I'm going to use some of this parafilm, cut some squares. Just going to put some on top here and put some on test tube B. And then I'm going to shake. So we shake for 30 minutes or 30 seconds, and then we're gonna just stop. So I'll show you test tube A and B here. And we can compare them right after I've been shaking. So test tube B, this one has bile salts in it. So we're now gonna observe what happens over the next couple of minutes and see if we can get the oil and the water to separate out, which we would expect to happen in A, but let's see what happens in test tube B where we added the bile salts.
Okay, so it's almost 1 minute. So 1 minute, let's take a look at them again.
All right, so we waited a minute. You can see the water and the oil they're starting to separate here in test tube A. In test tube B, we have some foam and still an emulsification situation. So we'll wait another minute and take a look at what happens then. All right, so we're going to take a look at our test tubes now after 2 minutes. You can see at the bottom of test tube B, we have a little water down here. It's colored a little brown, yellowish, because there are bile salts also in this water. And you can see that the test tube A, we have the water layer and the oil layer coming back. So we'll wait another minute and take a look at what happens after another minute. All right, so after 3 minutes, we can see that we have water and oil separating in test tube A, and in test tube B, we see an emulsification layer here in the middle, and then water down at the bottom, and then we have some foam up here at the top.
Right, so you can see it's pretty clearly the oil is coming back on top, the water is on the bottom. Here, this whole zone in here, which you can see is fatter than the layer of oil or water. This is a mix that is intermingled, little fat droplets that stay suspended in the water. You know, so there is some water just on the bottom here, but most of this is this kind of what we call emulsion of tiny little droplets that are staying separated from each other, kind of suspended within the water. So that makes sense. You know, when you interpret the results for the next part of the lab, it is important to remember that the cream that we're starting with is already an emulsion to a degree, because it's like kind of fat and water together that are kind of staying in this kind of equilibrium. So when we add the bile salts, it's going to further emulsify it, but it's starting out already kind of emulsified. So you're going to probably not be that shocked that the lipase can actually be relatively effective even without the bile salts, but the bile salts will help. So. All right, so the last part, we are gonna, let's see, the time is, we're all totally cool. We are now gonna watch the last video, which is gonna be looking at part 3, where we actually have the test tubes with the cream and the lipase and the bile salts. And then once we've done with that, we can go to the results.
All right, so next we're gonna do our experiment where we're using litmus cream, which is just a heavy whipping cream where we mix some litmus powder in. And so this is a pH indicator. So it's at the start here, it's blue and it turns pink if you add acid. And so I'm gonna illustrate that for you by adding some hydrochloric acid into this test tube, and we'll see how it should turn pink. And you can see it's indicating that we added an acid. All right, so we're going to do the next part of this experiment. So what I have here are 4 test tubes clearly labeled. It's so important that you keep track of which test tube is what getting what treatment. So next, I'm going to add 3 milliliters of litmus cream into each test tube. And so I'm going to try to be efficient here and just do it with 1 pipette, stick it all the way down to try to avoid getting anything on the side of the test tube, and then slowly add 3 milliliters, and then move over to the next and add another 3. And the next. And then I'm going to have to refill. So it helps not go all the way to the bottom of the screen because it could be some particles that could clog the nozzles, but let's see. Oops. I'm going to do the last one. And there we go. Great. So next, I'm just going to follow my table and add the various treatments. So for the first one, we have a pancreatin solution here, and I'm going to add 3 mL of that to test tube 1.
Here again, pancreatin is just a particular lipase that is produced by the pancreas. So it gets the name. We're going to see that pancreas makes lots of different kinds of enzymes, not just lipases, but pancreatin is the specific name for the enzyme that is a lipase made by the pancreas.
Okay, and I'm also going to have 3 mL to test tube 3. And then the next, test tube 2, I'm going to add a pinch of Biosalt and some water, 3 ml. And then test tube 4, just 3 ml water. So the trick here is to not get the test tube wet on the walls here. So when you add the powder, it all just gets stuck on the wall. So I was trying to be careful with that. So the next, I'm just going to add my pinch of Biosalt to test tube 2.
Another thing you don't get to experience is the bile salts are really stinky. So when you're doing this, it's not just like adding, it's like, so just to try to give you some like more visceral experience of what it would have been like to be in the lab, you would have probably kind of thought like, this does not smell good.
And we're going to try to get it all mixed in too. So next I'm going to add water to test tube 2 and 4. And a pinch of Biosalt into test tube 3. So next I'm going to try to get the Biosalt dissolved into these test tubes. Might have to use some paraffin. Use a stirring rod. Get it off the walls. And I am not cross-contaminating my samples. I'm using the clean side of the glass rod in this one. That's pretty good. All right, so next I'm going to walk these over to the water bath, and it's going to sit there for an hour at 37 degrees C. A little thought question, why 37? What does that have to do with our bodies and physiology?
Yeah, what is the answer there?
Our body
Our
temp?
body temperature.
Yeah, it's our body temperature in Celsius. So it'd be 97.3 if it was Fahrenheit.
All right, so the temperature is 37 degrees, and then I'm going to sit here for an hour. Okay, so here I am taking out my test tube. They've been in for an hour, and we'll just bring them over to our lab bench, and we're going to take a look at what's happened. All right, so your job now is to think about our pH indicator, what has happened to each tube in terms of pH and why that happened. And also look at the color and compare the different tubes. So I'll take out test tube 1. Here's test tube 1. we'll stick test tube 2 next to it. Let's see if I can hold them here. Test tube 3 and test tube 4. All right, so it does look like test tube 3 is a little bit more pink than test tube 1. And I think if you just look at each of the tube and write down the amount of digestion that has happened and think about what did we put in the tube and how does your result make sense in terms of the various treatments. All right.
Okie dokie. The one other thing that the lab manual says that we didn't show you here is it asks you to smell the different test tubes from the lipid digestion part. And what I can tell you, in fact, let's go to the next. What I can tell you is, and here you, in this picture, I kind of did the lighting so you could really see a little better. Test tube 3 was a bit more intense pink than test tube 1. Now, what does this tell us? What can I say? What do I know about test tube 1 and 3 compared to test tubes 2 and 4? Just from looking at the colors here.
It's more acidic.
They're more acidic, which means
lipids got broken down.
Which means that the lipids got broken down, whereas in test tube 4 and 2, there was not that breakdown because we don't see them turning pink like that. I can tell you that if you were putting your nose on top of them, test tubes 1 and 3 would smell like a baby vomited. So I was in this, so I actually asked one of the chemistry professors more about what's going on there. I guess the shorter chain fatty acids that tend to show up when there is fat breakdown tend to just be stinky. That's just the nature of them. So when you have these, if you have the fats are all connected up in triglycerides with their, it smells just like, it smells like cream, which is nice. But if you have these short chain fatty acids freed up, they smell like spoiled milk. But it's pretty dramatic how test tubes 1 and 3 smell. And 3 is the worst in terms of just smelling like, like spoiled milk. So.
What about test tube 2? You said the bile salts smell pretty bad by themselves, right?
Right.
Were you able to smell that with test tube 2?
But it's not this. It's that they smell bad, but they don't smell. They especially when you just put a tiny little pinch in with the cream. It's not the same thing as smelling spoiled milk in the other ones. So, yeah, my experience at that in sniffing the 4 tubes is 2 and 4 aren't bad. I'm trying to remember if I really noticed the bile salts in 2. I don't remember if I remember, or I don't remember if we did or I did. I can say for sure the experience of 1 and 3 is like, so here are the results. You can use these for your write-up. Here's Lugol's and Benedict's from the part 1. Here are the lipid just color assay from part 3. Like I said, unfortunately, part 2, there wasn't really anything to give you a good picture of. And then the last piece here is some results from last year, which I think are useful. Because like I said, in general, you don't want to just look at one experiment and just say, I know the answer. If you have a lot of groups that do the same thing and get the same answer, then you trust it more. Actually, I want to open. So what this is just to kind of give you a sense of many groups doing the same thing. So for part one, these are 6 different groups. And looking at carbohydrates as assessed by Lugol's, test tube 1, everybody said full on carbohydrate, no maltose. Again, if we look at our controls also, full on carbs, no maltose. For the maltose, no carbs, full maltose. For test tube 2, most people said no starch left at all. One group said a little bit of starch, but in general, it's pretty clear starch is disappearing and maltose is appearing. Whereas in test tube 3, it's pretty clear. Even the people who thought like, oh, maybe not as much starch, they still didn't see maltose showing up. So that's telling me that there was no breakdown. So this is nice. Basically, 6 groups did the same thing and all kind of pretty consistently got what we saw in the original example that I showed you in the YouTube video. This particular lab worked a little better for the protein. Here we actually did have this group that had the pepsin and acid seem to have the most breakdown. So if you look at the data from this actually 2019 actually does, which tube would you have expected to have the best breakdown for the protein digestion part?
Tube 3,
Why is that?
because of the presence of acid, allows pepsin to do its job. I actually wondered if we'd see a little bit in tube 2, also just from acid breaking down the proteins.
Right. In general, though, acid, first, so it's a pretty weak acid. This is much weaker than the concentrated hydrochloric. So it's more about adjusting pH, but it's not really gonna be like kind of ripping anything apart. So lipid 1, we can see, basically it's very similar to what we saw in the picture I just showed you. Tubes 2 and 4 really just stayed blue. Tube 1 turned pink. There was some digestion, but it was even pinker, more digestion in tube 3. So again, use this as more support. You can have in your discussion, you can discuss, the particular results from, when I did this with Tina and Becky, but then this is important to bring in to corroborate and say, like, these results are consistent with the results when many groups, did the same procedure. Because that's something I want you to walk. In fact, especially this is our last lab. Kind of maybe take home messages even about, about scientific studies. One is the objective, have a clear idea of what you're actually trying to figure out. What is the question? Right? Because that makes a big difference to actually get an answer, is if you actually have a clear idea of what exactly are you trying to figure out here. And then the other thing is, if you do an experiment or you check something out, if you just do it once and you get some information, that's called anecdotal evidence, right? That's like, okay, that's interesting. It's informative. But if you really want to have more confidence that it's really the real story, you need to either do it again and get the same result, or have other groups doing the same thing and getting the same results. And then you can start trusting more that, oh, this is more than just like, some randomness that I happen to look at in this moment.